Hi everyone, my name is Vazquez and I am taking personal genomics with Dr. Prescott at the University of New Hampshire, Manchester. I have decided to make this PowerPoint to be able to show you what I have taken back from genomics and be able to explain to you what I found that was most helpful. So I did decide to talk about a little bit about an intro to genomics, just because going into this class I really didn't know what I was going to study, and this really, you need to really understand have a basic concept understanding of these processes in order to understand genomics itself. So let's get started. What is a genome? It is the entire sum of genetic compounds of any living organism. In other words, it is all of the genetic stuff that defines an individual. But don't get confused, because the media often refers to the genome as a series of genes unique to an individual, but the genome is much more than that. I'm just telling this because the media tends to simplify things. We all know that, and I just want you to understand that this is way more complex than anybody could ever explain. So you can come into genomics thinking that it's going to be straightforward. Genes, DNA, it's tricky, it's complex. So this is why I want you to push aside the simplified views that the media may have given you. Only less than 2% of the human genome is known to code for genes. The remaining 98% is considered junk DNA, for now at least. The human genome is something that is currently being vastly studied. Scientists are learning new information every single day. It's very important. This is just a little breakdown of the human genome. So you're going to see on the left side, it's going to be the 2% that we already know, all those genes that we code for. And on the right side, you're just going to see that 98% of junk DNA. Just a little image to kind of give you a visual. This next thing is a very important concept of DNA, the central dogma. So it is really important to understand this in order to be able to understand genomics. It is the process of how DNA is transcribed and translated into polypeptide chains. So first we have DNA here, and it's going to go through transcription, which is when this segment of DNA is copied into mRNA, which you can see right here. So DNA double helix, mRNA single helix. After that, mRNA is translated into an amino acid sequence or a polypeptide chain. Here, it's going to make the proteins or the amino acids we need to express genes, to code for different functions of life. So something that I really want you to focus on is those little letters that you see here, the A, T, G, A. These are your nucleotides, which are very important because one single change in a nucleotide can actually give you a different protein, so a different function. So it really can change a lot, and this is where a lot of mutations and problems come from. So how does a genome work? Having a genome sequence is inconsequential because the gene is not expressed. Biological effects inside and outside the cell will only occur if the gene is expressed. Gene expression is very, very important, and also gene regulation. A gene is expressed through transcription and translation, and the final result will make a single protein. A cell must be able to precisely express thousands of genes to support a single cell. So this, again, is why the concept of central dogma is so important, and that's why I want to focus on. Right here in the picture, you can actually see in the bottom that there's 3 different proteins, A, B, and C, and how they're all different. And a single nucleotide change can really affect the function of each single protein and its function. So this leads me into gene regulation, which is the process of controlling which genes in a cell's DNA are expressed to make functional products like proteins. So this actually leads into talking about transcription factors, which are a set of regulatory proteins that allow the protein to attach to and bind to specific regions of the genome. These transcription factors really help us understand different regions of the genome, just because we are able to look further into them and understand them. But however, the biology of gene regulation is still something that is being researched. Just like this field, you're growing with the field, you're really understanding things. Some things you're not going to understand because people don't know the answer to them yet, which is really interesting because it's something new and something that you want to learn more about, and you can actually do research yourself. And so that's one of the reasons why I found this class very interesting. Right on the left side is a picture of the levels of protein. So you have primary, secondary, tertiary, etc. As the protein keeps getting more and more complex. So these are what you are going to call your insulin, your hemoglobin, that are very important for human function. So that's why this whole concept is important, because it's going to lead to these crucial proteins. So here we have the human epigenome. So the epigenome is an abundance of chemical compounds that can tell the genome what to do. The epigenomic compounds attach to DNA and modify its functions. These compounds mark the genome but do not change the sequence of DNA. However, they change the way the cells use the instructions of DNA. So very manipulative compounds here. But when we talk about these compounds, we have to talk about two different factors that affect that, which are genomic imprinting, which is, despite the fact that an individual has two different genes, one from mom, one from dad, not both will be expressed. So this just becomes a conflict because both of them are supposed to be expressed, but one isn't. So you're missing a gene there, which can be complicated in the long run. But it is very rare. However, it does happen. Another thing that we need to talk about is environmental factors. The environment can work through epigenetics mechanisms to alter human biology. So the environment around us really affects everything we do, and it's even up to our DNA. Genetic variation. This describes the observed variability in genetic and genomic characteristics between individuals and population. So basically, me and you are different, me and a rat are different. Genetic variation. Polymorphism. This term describes the genetic variability that can be found along the genome at a particular locus. So, a single nucleotide can actually really cause a lot of variation within a population, because that can cause different expressions of genes and different proteins. So humans have about 1% variability of each other. So that is literally just one nucleotide. Polymorphisms come into existence by mutations that persisted throughout history and spread throughout the population. Again, evolution. Favorite gene is going to keep getting expressed. This mutation is going to keep going down the line. And these mutations can be caused by errors in DNA replication, so that nucleotide change, radiation, mutagenic toxins, and many more. Polymorphisms are often inherited together due to linkage disequilibrium. As the mutation arises, they are inherited together with other nearby alleles. So right here, you just kind of see a picture of how they are together. And linked. So linkage disequilibrium refers to the non-random association of alleles at two or more loci. It's just a very important mutation. This brings up an important concept of genomics called SNPs, single nucleotide polymorphism. So this is important because most of the data that you're going to see in the future is going to be SNP data. And at first I didn't understand it, but now I do. So it is really important, because if you see all this data, you won't even know what it means unless you know what an SNP is. And it goes all the way back to those little nucleotides that I was telling you about. And these are the most common type of genetic variation among people. Each SNP represents a difference in a single DNA building block, which is a nucleotide. like I just said. So to wrap this up, I just wanted to show a little bit about the Human Genome Project. I gave a picture here with a timeline. So it did take scientists a couple years to be able to sequence a whole human genome, and it's something that it has been recent in the last decade. So that's why we're learning more about it, and that's why it's becoming such a big and growing field now, because we're understanding more things, and it's really something great. Thank you so much for listening, and I hope you can take some of this and apply it to your personal genomics class. Here are some references.